all to yet another exciting seminar from ATI. We have Gayle Piat today. Before that, we acknowledge that the University of Alberta is located on Treaty 6 territory and respect the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. And special thanks to Estellas for their continued support of our ATI seminar series. Today we have Gayle Piat. I'm sure all of us know her very well over the years. She has over 30 years of experience in the life sciences, specializing in good laboratory practice and good manufacturing practice. She is the director of the Alberta Cell Therapy Manufacturing, located at the University of Alberta, where she oversees all GMP cell therapy manufacturing for clinical trials. She's also a registered quality assurance professional in GLP and holds a regulatory affairs certification through the Regulatory Affairs Professional Society. Her talk today is about transforming innovation into therapies, manufacturing for clinical trials. The floor is yours, Gayle.
And thank you to the Alberta Transplant Institute for inviting me here today to speak. As mentioned, I'm Director of Alberta Cell Therapy Manufacturing, or ACTM, located here at the University of Alberta. ACTM manufactures products for cell therapy clinical trials, and more recently we have started aseptic fill finish as well, and I'll be talking a little bit about that later on. So the title of today's talk is Transforming Innovation into Therapies, Manufacturing for Clinical Trials. And what I'd like to do is to walk you through how we take discoveries from the lab, exciting, promising science, and actually turn them into therapies that can be tested in humans in clinical trials. So along the way, I'll be highlighting the role of good manufacturing practice, or GMP, the stages of clinical trial manufacturing, some of the challenges that we run into, and the strategies that help us succeed. First, I'd just like to say that I don't have any conflict of interest, and I definitely will not be discussing any off-label use of drugs and devices. So we've all heard the term valley of death in translational research, and that's the gap between an exciting new discovery in the lab and then an actual therapy reaching patients. Many of these great ideas never make it out of the lab, not because the science is wrong, but because the path to manufacturing and regulatory approval is so difficult, and not to mention expensive, of course. So this valley is where innovation often stalls, and manufacturing, or specifically good manufacturing, helps to build that bridge across the valley. And without it, a lot of promising therapies can fall short. So oftentimes I will get a researcher that will come to me and say, we had Eureka, we have a great new discovery, it's working, and we are ready to go to the clinic. And I get to be the bearer of bad news to say not quite. Oftentimes people aren't aware of all of the steps that have to go into actually getting into that patient. And some of it is process development, actually converting the process into a GMP ready process. There's a lot of work that has to be done and a lot of steps that have to be done to make that happen. So it's called tech transfer when you're going from a lab to a contract development manufacturing organization, or CDMO is the acronym for that. And so that's what ACTM serves as. And at some point we'll take that process and we'll lock it down. And that's when you want to start manufacturing product to do GLP safety studies or toxicology studies. And you want to make sure that it's going to be an equivalent process to what you're actually going to be testing in people as well. So you don't want to change the process too much after that. And then we do 3 dry runs essentially, or engineering runs that aren't going into people yet, but they're close to being GMP compliant at that point. because you want to demonstrate to the regulator that you can run 3 batches in a reproducible manner and that pass all of your release testing, quality control testing. Oftentimes those batches are used for stability studies, and all of this information on the slide is then gathered into a clinical trial application and submitted to the regulators to get approval to go forward for your clinical trial. So this is kind of a different snapshot of what it looks like to go from R&D or discovery to a commercial product. So you're going through the animal safety studies, following good laboratory practice, you're following good manufacturing practice and good clinical practice throughout your phase 1, phase 2, phase 3 clinical trials. And then finally, you're getting marketed approval where you're at full commercial GMP at that point. So by the end of this session, I'd really like you to walk away with 3 things. I'd like you to have a good understanding of the role of GMP in clinical trial manufacturing and why it's not just a regulatory checkbox, but truly the foundation for patient safety and trust. An overview of the key stages that we go through from process design all the way to product release, and a look at some of the common challenges and the strategies that we use to balance innovation with safety and compliance. So GMP is really about 3 things: consistency, traceability, and reproducibility. So simply stated, it's you say what you do what you say, and then you prove it. So GMP in a clinical trial setting is a little bit different from full commercial GMP, because clinical GMP is phase appropriate, meaning that early phases allow for more flexibility and learning with good manufacturing practice and compliance with the regulations. But by phase 3, everything is tightly locked down and there's no changes to the process at all at that point. So regardless of the phase, GMP ensures one thing overall, and that is patient safety. That is the goal. And that's why regulators, clinicians, and patients trust products made under GMP. So if you want a bit of a mental picture, think about cooking. So at home, you might add a pinch of salt here or there, some extra spice to change a dish that you're perfecting, and that's research. And in a restaurant, you want more consistency, and you would want the same dish coming out the same time and tasting the same each time, and that's GMP. So moving from a research lab into a GMP environment is a huge cultural shift. Research thrives on flexibility and improvisation, sometimes a little bit of chaos maybe. And GMP thrives on control and documentation and reproducibility. Every step is defined and recorded, and it must be repeatable. If it wasn't written down, it didn't happen. So it makes me wonder about some of my workouts that I do at the gym. So a case study would be maybe if you had a promising CAR T therapy facing an 18-month delay because researchers discover that the raw materials that they chose for their process aren't available in GMP grade, they're only available in research grade. And then you have to go back and do a bunch of validation and comparabilities studies once you make that change. And so not planning for GMP in the beginning or beginning with the end in mind can really cause a lot of delays and significant costs. So the lesson is simple. Decisions that you make in the lab, like which raw materials you choose, can have major consequences later on and painful surprises in some cases. So here's kind of a visual shot of thinking about what it looks like in a research lab. And this is a shot from our clean rooms here at ACTM and what the process looks like and what a day in the life looks like for our production personnel that are in the clean room doing the process. Everything takes much longer in the clean room than it does in the research lab. And one of the reasons is how they have to dress. It takes a while to get dressed up like this to go into the clean room, and you don't want to go out and numerous times if you forget things, and all of the documentation and verification steps that have to go on as well along the way. So process and analytical development are the foundation of how GMP manufacturing starts out. So first we have to define what we call critical quality attributes. And these are the things that ensure that the product is safe and effective. And that they're making sure that we're looking at safety endpoints, potency endpoints, purity, and things that would directly impact the patient outcomes and the therapeutic efficacy. And then we're also identifying critical process parameters, and those are the steps in the process that can have the biggest impact on these critical quality attributes. And we want to test the process as we go along so that we can get a feeling of how much our process is in control early before we get to the end. We don't want to wait until the very end to find out whether our process worked. So we monitor and do QC testing as we go. And then finally, we develop analytical assays. And analytical assays are often forgot about during the process development, but these are really key. And we want to make sure that they're reliable and that they're scalable. In phase one, a lot of times these are just fit for purpose, meaning that they're not fully validated assays, except for maybe the safety, like sterility and endotoxin mycoplasma. But the efficacy tested for potency might not be fully validated in phase one, and often isn't. But by phase three, every QC test that you're doing at your release testing should be all fully validated. So that's an example of phase appropriateness for GMP, where in early phases, things might not be fully validated, but by phase three and commercial, they are. So this stage is about building robustness into the process. And if the foundation isn't strong, then everything downstream, including QC, manufacturing, and regulatory approval, can be at risk. So tech transfer. Tech transfer is when that process is then moved from the development lab into the GMP clean room. And it's not just the process that moves, but it's the knowledge that has to transfer as well. So this means standard operating procedures and detailed documentation, lots of staff training, and equipment qualification, meaning, and equipment qualification, you can see here the acronym IQOQ-PQ, that stands for installation qualification, operational qualification, and performance qualification. So we are testing that equipment like crazy before we use it in a process to make sure that it operates as intended. And before the first GMP run, we always do a dry run or a dress rehearsal to make sure that everything is ready, the process, the equipment, and the people. So I mentioned standard operating procedures. This is just a snapshot of our filing cabinet here at ACTM. We have a lot of SOPs. We have SOPs on how to write an SOP. We have SOPs on how we train our staff. We have SOPs on how we document things, how we operate our equipment, how we do QC assays. Essentially, everything that we do has a standard operating procedure, and we have to keep the historical versions of all of our SOPs. And then here's just a little video of thinking about some of the equipment monitoring that we do during the process. Is that we're doing environmental monitoring, and we're testing the particles in the biological safety cabinet and in the clean room. We're doing active air monitoring here to check for colony forming units to make sure that everything is under control. And we do this routinely, and also we do it while we're doing the process, so that we know that when we were actually manufacturing a process, our clean rooms and our equipment, our biological safety cabinets were operating properly, and that our staff were performing the procedures with aseptic technique. Now, this is, I mentioned equipment qualification, the IQ, OQ, PQ. This is, it's not it's insignificant amount of work to do this. This is for one of our autoclaves. So all of our, it's a fair amount of protocol that has to be gone through, testing alarms, testing the temperature in the autoclave, testing that it actually does sterilize something by doing the performance qualification. So all of this work has to be done before we even start producing a product. So now manufacturing execution. This is where the rubber actually meets the road. Raw materials that we use in manufacturing has to be qualified, it has to be traceable, it has to be consistent. And this is often the Achilles heel of the process. If we change suppliers, if we have different lot numbers coming in, we have to make sure that we actually know what we're putting into that process, because it's especially if those ingredients are going to end up in the final product. That's extremely important. We have to know that they're sterile and that they're appropriate for use. Aseptic technique is non-negotiable. Cell therapies in particular, we can't sterilize them at the end, like you can with small molecules, where you might be able to heat sterilize a product or sterile filter it before it goes into the patient. With cell therapies, we have to rely on aseptic technique or closed processing to ensure that our end product is sterile. Batch records must capture every detail. If it, as I said before, if it isn't written down, it didn't happen. And we have to have complete traceability. You think about an autologous product like CAR T, where you have the patient's cells as being the starting material, and then the patient receives those same cells back. Chain of custody is absolutely critical. Each batch belongs to one patient, and mixing them up could be catastrophic. So quality control and testing. So quality control is really the gatekeeper. So we do raw material testing, and in early phases, sometimes the raw material testing is simply looking at the certificate of analysis and making sure that it's appropriate, and making sure, looking at every bottle of media and making sure that it's intact and isn't leaking and it's the right color, etc. But in later stages of manufacturing, you might actually be taking a sample of the raw materials and doing quality control testing on it to ensure that it passes before using it in manufacturing. Because you want to catch those things and the problems with any of the raw materials early on. And you want to make sure that the product meets specification. And then you've got in-process control, so that's real-time monitoring and testing during manufacturing. So you might be doing some cell counts, for instance, during your process. And then you've got final release testing. So that's when you're doing your comprehensive analytical testing, including sterility and endotoxin, mycoplasma, identity, purity, potency, before you're releasing that product to the patients. And cell therapies highlight a particular challenge because they have shelf life, sometimes of just a few hours. So QC must be fast and efficient. And sometimes you have conditional release where you might be releasing the product without the final sterility sample, and you might take a sterility a few days before you harvest the cells, have a actual result for that product release step, but not for your final product. And in that case, if there was something that came up, we have to have a call with the physician and let them know that, okay, there is an issue, and then they might have to start antibiotics, etc., and treatments. Biologics are more stable, and but sometimes they require a lot more extensive testing, perhaps, than cell therapy products might. So every product is different and every process is different. So then we have batch review and product release. And so this is where quality assurance, an independent quality assurance group, does a comprehensive evaluation of all of the records that were done. It could be deviations that happened during the process. It could be corrective actions. It could be training logs of the staff that were actually working on the process. It could be all of the batch records itself, the analytical testing that was done. But essentially, nothing moves forward until QA signs off on it. Regulators don't just inspect the product, they inspect the process that made it. So documentation is as critical as the process itself. And documentation integrity is very important. So making sure that the batch records are accurate and that they meet the regulatory requirements for traceability. And then product release happens only after all this happens, which makes CAR-T of a fresh product very tricky because we have to do all of those documentation verifications and corrections very quickly so that we can get the product to the patient. And just another little snapshot of one of our documents here. This is a batch record from a CAR-T patient. So all of this paper that you see here is for one patient. So essentially everything that we do in the process steps has to be written down. There's a lot of supplementary documentation that goes along with this, like equipment records and raw material receipt and inspections and quality assurance testing and all that sort of thing. So the record looks a lot bigger than this, but this is the actual batch record if you haven't had the pleasure of seeing one before. So some of the key challenges that we have when we're looking at manufacturing, and there are plenty, especially I think in clinical trial manufacturing, because you often are doing it for the first time in a phase 1. And we're doing small batches often for cell therapy or for phase 1. And with small batches, you can have a lot of variability. If you think about making batches of cookies and you're making one cookie over, and that's your batch, and you're trying to make all those cookies over 3 months look exactly the same as one another and taste the same. It's the same with us, and sometimes our starting material is different. So if you have an autologous product where you have different patients as the starting material, one patient's cells may act differently than another patient's cells in that process. Or even for allogenic, if we have a bone marrow donor, we might be able to make several products from that one donor, but then that donor bank eventually runs out and you have to start a new bank. That patient's bone marrow may be very different than patient number 1 or donor number 1 was. So that is, sorry, I'm just confirming something here. So that is very important when you're looking at variability, process that evolve as trials progress, because you learn things as you go along, both in the process and also the clinical outcomes. Raw material shortages and variability, that happens quite often, especially in early phase trials, because it might be a really unique raw material that's used for a process, and the manufacturer may not have a lot of experience in manufacturing that product, and so there could be a lot of variability or changes with the raw material. So it's a good idea to have good communication with vendors to know when they're going to change something, and if they do change it, what they change. Supply chain risks. It's dangerous to rely on a single supplier, because if they go out of business or they run out of stock and you don't have backup, then you run into delays. But in some cases, it's difficult to find a backup supplier because it may be a novel process and a novel raw material that you can't find another, especially a GMP supplier. And then there's always the constant tension between speed and compliance. Time is money, and often there's a lot of pressure to move quickly through a clinical trial and keep costs down. But when you go too fast, sometimes you can increase your risks and have issues with compliance. So it's knowing a tricky balance to keep all of that in mind. So a case example can be a viral vector program, depends on one plasmid supplier, and then when that supplier changes their process, the new plasmids don't work as expected, and then the company has to pause, qualify a new supplier, prove comparability, and then it could delay their trial by nearly a year. So it's a good thing to kind of do a risk assessment from the beginning. So how do we deal with these challenges? Well, some of the things that we do is, as I mentioned, there's phase appropriate GMP. So we do apply that in early phases, and there's a lot stricter controls as trials advance. But when you're looking at how do we choose what we consider as, you know, hard, fast rules to follow, the GMPs, or how do you choose what is phase appropriate, is that what we end up doing is a lot of risk-based decisions. So we use tools like FEMA and other risk assessment tools and look at the quality attributes and focus so that we can focus our resources on the highest impact areas. And so we don't paint everything with the same brush, that we really make good decisions when we're choosing what we're going to spend our time on. And then there's a lot of quality culture and training that goes into running a GMP facility, and it's about fostering that commitment to quality and accountability for a good product at the end. So the best SOPs in the world won't help if people aren't following them. So people really have to buy into that culture. And then automation and digital tools are good to look at reducing human error and helping with efficiency and reproducibility. And so one thing that we've looked at our facility with automation is using the Milteney Prodigy for a CAR-T project. So that's using a closed automated cell therapy manufacturing piece of equipment, and then also with vial filling. So we used to fill vials, drug vials for clinical trials, either manually or with a semi-automated system, and we've recently received funding to get a robotic system, and I have a video of that at the end that I can show you hot off the press. So errors can dramatically drop, sterility can improve, and staff workload is eased when you look at some of these automation tools. And then early regulatory engagement is super important to decrease your challenges and surprises along the way, and having pre-CTA meetings with Health Canada and getting some of your questions answered early is really important to build confidence and alignment with the regulators. So just have a little quick video here. This is the Prodigy. So this is our CAR-T product that we run for cross cancer. We've treated over 32 patients. And so as you could see in this video, everything is all in with peristaltic pumps and closed tubing sets. And what you see here is the patient's cells going into this little centrifugation system that also acts as the incubator, where we then harvest the sample at the end. So this is a shot of, I mentioned, aseptically filling vials. And so this is what we used to do, and had a semi-automated system where we had a closed vial from Aseptic Technologies, and someone would be sitting here in this biological safety cabinet taking a vial one by one, and putting it on the stand, pushing a button, the needle comes down, pierces the septum, dispenses the liquid, comes back up, and then you take that vial and put it in this box here, and there's a laser, it's a laser box. So a laser would hit the top of the septum and seal the hole that the needle made, and then put a cap on it. So one by one, filling these vials, or using a repeater pipette and manually stoppering and crimping is another technique. But so these biological safety cabinets are in a grade B clean room, and everyone's dressed head to toe in Tyvek suits and performing aseptic techniques. So you can see that there are some, even though you're doing aseptic technique, you still have a lot of interactions with employees at that point. So what we've decided to go to is in a robotic system. So this is a shot of our robotic system that we had special designed in Italy from a company called Steriline. And it took a lot of time to customize this piece of equipment to fit in our existing clean room. And so previously we were able to fill about 500 vials a day. This piece of equipment will fill 700 vials per hour, and it will fill pre-filled syringes as well. So it is currently at the port in Montreal. It's made its way across the ocean, and it's waiting to get on the rails to come over to Edmonton soon. And so then it's going to be installed in October, and then we have a lot of validation and everything to do to get it ready for filling vials and syringes, but we expect it to be up and running probably mid to late 2026. So I'm just going to run through a little quick video. This is the first time publicly that anybody's seeing this video, so it's pretty exciting. But this is what it's going to end up being able to do for us. So there's no sound on this video, but here you can see it picking up a nest of vials from a tub, and then the robotic arm will start picking up the vials and moving it to the filling station. And it's going to start. Sorry, my phone was ringing. It's filling the vials one by one, and as it's filling, it's weighing the vials so that you know that you have the correct amount in each vial. So if it is underfilling, it will add a couple drops of drug product back into the vial until it's at the proper weight. So there's no wastage or no rejects in this case. So then the next robotic arm will take it to the stopper station. This is where it's doing the 100% weight checking. And so you'll see in the next shot here where it's going to add a couple extra drops because it filled under. So some filling equipment you would actually, it's weighed at the end, or it's just some of them are weighed. So at the very end, when you're weighing all the vials, that's when you may have to reject them if it didn't fill what you wanted it to fill. So here it's stoppering. And it's also got a camera on the robotic arm, so it can tell if the stopper wasn't placed properly. So again, some other pieces of equipment, it would actually reject it. It wouldn't know that it wasn't stoppered until later on. But here it detects it, and so then it restoppers it. And so then we have a good vial. This is very important for very expensive products like cell therapies and gene therapies where every drop counts. So this whole system has been designed with that in mind. So we'll be filling phase 1 and phase 2 clinical trials, and we can fill vaccines, monoclonal antibodies, small molecules, cell therapies, saline, anything that needs to be filled aseptically. and also recapping. So again, you can see if you look really quickly here at, it's very fast, but it puts the green cap on, and if the cap is missing, it'll recap it. So then when it's all finished, it moves it over to the tub, and then it takes it out on a conveyor belt, and it's finished. So there's 120 vials filled in an hour in what used to take us more than all day. So just to wrap up, some key takeaways here are three things that I'd like, hopefully, that you took away is that GMP is the foundation of trust in clinical trial manufacturing. That the manufacturing journey goes from process design all the way through to product release, and that the challenges can be managed with strong strategies, risk-based approaches, and early planning, so beginning with the end in mind. And so GMP may not be glamorous, but without it, innovation sometimes is a tricky path. So now I'd love to hear your questions, yeah, any questions that you have, I'm all ears.
Okay, got a question from Donna. She says, how has this new equipment improved your lab?
So as far as the Meltony Prodigy for CAR T, I think that we've done a lot of cell therapy manufacturing that's been manual. So the staff are in the clean room and gowned up in the full Tyvek suits for many hours, not drinking a lot of coffee before they go in there, of course, because you can't just come and go. And so I think the automated equipment allows us to operate in a lower grade of clean room because it's closed. And so the staff then can wear just a lab coat and a bouffant and booties and gloves. So it's a lot more comfortable for the staff. It's not as hands-off as maybe the vendors would like project when they're selling a piece of equipment. There's still a lot of work to do. Like when we're harvesting CAR T, there's 6 staff members in the clean room doing that process for about 6 hours and doing all the quality control testing. So there is still a lot of work for the staff to do. So it's not putting people out of work, so to speak, but it is helping create a safer and more reproducible product.
And we have a question from Tatsuya here. He wants to know about your operational environmental monitoring process. Like you did mention about your readiness, like at-rest EM you conduct in the BSCs and the facilities. Like what about operational? Like do you have any process in place where, for example, you're doing a CAR T cell production, you do an operational for that particular process? And if it's like a biologic manufacturing, do you have a separate kind of operational, or it's like standardized and
For environmental monitoring?
yes, yeah.
Yeah. Yes, our environmental monitoring is very process specific because we look at the critical parts of the process and where are the highest risks. So that's where we would choose to do more sampling. Let's say where we're sampling the staff after they've done a critical process and sampling their gloves, etc., to make sure that they haven't contaminated their gloves, for instance. So that is really done on a case-by-case, process-by-process basis. Our routine monitoring is pretty specific to the room and the configuration of the room, etc., but the process environmental monitoring is process specific.
Yeah, I do have a question about like for manufacturing, there are human and or animal derived materials for cell and gene therapy products. So there's a clear distinction for as a GMP facility. Do you need to, take care of any special considerations depending on the source? and like flexible with your process in that regards? Can you just shed some light on it?
For sure, yes. All of our raw materials, as I mentioned, have to be reviewed for GMP compliance before they're used, and the vendor assessment as well. And part of that assessment is looking at is there any animal derived products? Has even when you look at plastics, has there been any point during the manufacturing of that plastic where it could have come in contact with any animal ingredients? And then you need to know the source of where those animals could have been, etc. So you're really understanding your raw material. If something does have an animal ingredient in it, then you really have to dig deeper and say, okay, what is the source, and get regulatory approval and look at adventitious agent testing, etc. So the first choice is no animal ingredients. That's always the easiest. And if that isn't an option, then you have to dig deeper and see what the risks are. There's a really good guidance document um, put out by ICH on use on raw materials, and there's different risk classifications. So the lowest risk would be if something has a DIN number, for instance, and then you know that it's already gone through all the approvals. It's got a DIN number, and then it goes all the way to, okay, what if you can't source a GMP material? What do you have to consider then, and what kind of additional testing might you have to do to get regulatory approval to use that in a clinical trial?
Based on that, like, do you like foresee or already have experienced any kind of new or emerging risks in this in clinical trial manufacturing you can think of?
I think the risks really are um, the sourcing of materials. Like,
Sourcing.
if you're in the middle of a clinical trial and all of a sudden one of your reagents, you can't get them, there's a shortage. We've run into that where there might be something that is used as a drug, a clinical drug, and then it's used in manufacturing. Well, the priority is going to be that that continues to be going to patients as a drug and not for, a raw material. So shortages are really key. And
Mm.
also maybe the manufacturer, and these aren't new risks per se, but they are things to keep in mind. And also that, companies go out of business, and we've got tariff situations now. So
Mm.
cost can all of a sudden go up dramatically. And if that hasn't been taken into account in the budgeting, then and shipping. If it's a critical ingredient, you can have
Yeah.
shipping issues where maybe the temperature wasn't controlled. So making sure, do you have a process for monitoring that shipment? How is it stored when you do get it? So not new issues so much, but just continual issues.
Mm. Anyone else have a? Yeah, the other thing I was wanted to know from you that let's say you do this small batches of phase one, phase two manufacturing for a product. Uh, what lessons can you, users take away when they go for a larger commercial level scale up? Like what? How can you guide them? Okay, because at ACTM we are involved with like a relatively smaller batch of productions.
Yeah.
So when they make the next jump, like what key lessons do you think they should or you give to them like from your experience? Like can you say something about that?
Yeah, scale up is always so difficult. And I think there hasn't been a lot of experience going from phase two to phase three
Mhm.
in Canada in cell
Mhm.
therapy. So I think it's really engaging with a manufacturing facility that does phase three and commercial early on,
Mhm.
the same as
Okay.
we like. our clients to come to us early on before they're even ready for phase 1 or phase 2, because I think the sooner that people start communicating, like even one manufacturing facility to another, do they have similar equipment? Do they have, do they have the same type of quality control instrumentation that we have? All of those things are going to be important for transferring a process from one facility to another. And scale up is you're back to that process development step. And you're back to figuring out, can you source the raw material maybe in a different volume? So we used to buy bottles of media. Can you now, does that vendor now supply it in bags that might be more appropriate for a larger scale? So those are some of the things it's never too early to start thinking about, because that comes back to selecting vendors of your raw materials. And are they going to be able to scale up with you, or are you going to have to switch vendors?